Thank you so much for having me, Neil. My name's Helena Yu. I'm a thoracic medical oncologist at Memorial Sloan Kettering Cancer Center in New York, and I am very happy to be here presenting to you different relevant topics that relate to EGFR mutant lung cancer with exciting new data from 2024. So the first study that I will go over is the use of neoadjuvant osimertinib for early stage resectable disease. And so this was a really interesting investigator-initiated study that took patients with early stage, anywhere from stage 1 to 3A, EGFR mutant lung cancer that was amenable to surgical resection. All patients received a short course, 1 to 2 months, of single agent osimertinib without chemotherapy, and then underwent surgical resection if appropriate. The primary endpoint of the study was major pathologic response rate, which is an established endpoint in the early stage setting that correlates with survival and outcomes. And in total, 27 patients were accrued on this study. The demographics of patients accrued really was in line with what we see for EGFR mutant lung cancer, predominantly female, significant minority of patients that were Asian. And you can see from the clinical stage of the patients, the different stages were pretty well represented, but the majority did have stage 2 or 3 disease. And I think these results were really striking where the expectations were not met. The major pathologic response rate was surprisingly low at 15%. And if we put this into context, the major pathologic response rate that we've seen with chemotherapy is somewhere, or chemotherapy immunotherapy in the recently presented neoadjuvant studies really ranges from the 30 to 40% range. So this is far below what we see even for chemotherapy monotherapy. So this was striking in terms of results. So kind of the different conclusions I made from the data, of course, both the major pathologic response rate and the pathologic complete response rate were quite low. One of the questions that was raised is possibly, is this due to the short treatment time? The osimertinib was only given for 4 to 8 weeks, and so perhaps there needs to be more time to see a cytotoxic response. But I think what the main kind of conclusion is that chemotherapy may be an important component of neoadjuvant treatment for EGFR mutant lung cancer, where there is a clear benefit with cytoreduction that a short course of osimertinib does not seem to get. This is a little bit in contrast with what we've seen in terms of the incredible benefits of adjuvant osimertinib. So, based on the ADAURA study, 3 years of adjuvant osimertinib has been approved for some time, and we see really striking both disease-free survival benefit, but an overall survival benefit as well. And so, something about prolonged dosing of osimertinib does seem to lead to curing of more cancers and cytotoxic benefit, but interesting that we didn't see it in the neoadjuvant setting. I think the study that we're really awaiting is the NeoADAURA study, and so that study schema is up on the screen on the top, and it is a 3-arm study where we are, the study does enroll osimertinib monotherapy, but importantly, there's a chemotherapy and a chemotherapy osimertinib arm. So I think this is going to be the definitive study to see whether osimertinib adds to chemotherapy in that neoadjuvant setting. And I would just say, for these patients for the present time, not without having the NeoADAURA data, my standard of care practice is to do neoadjuvant chemotherapy and then surgery and then adjuvant osimertinib in the back end. But that might change with this NeoADAURA study. The other data that we saw last year was the ADAURA minimal residual disease analysis. And so the ADAURA study, as I just mentioned, is an adjuvant study looking at the 3 years of adjuvant osimertinib. And they did a lot of different correlative analyses, including using a tumor-informed minimal residual disease assay. And so that was tested prior to right at enrollment of the study, prior to surgery, after surgery, and then serially after surgery while on osimertinib. And you can see they have a really robust data set of more than 200 patients that had serial MRD analysis. I think importantly, on the top right, you can see the baseline MRD status, where the majority of patients were MRD negative after surgery, but prior to going on adjuvant osimertinib. And then you can see the MRD lead time. So this is how soon before radiologic or clinical progression was the MRD assay able to detect residual disease, and that lead time was about 5 months. And then finally, when looking at patients who received adjuvant osimertinib, it's interesting to note that most of the patients that had recurrent disease, either by MRD or clinical progression, actually had that after they completed their 3 years of adjuvant osimertinib. And so, again, stating that the recurrences happened predominantly after osimertinib was discontinued, and that also raises the question, should we be giving longer or indefinite osimertinib for these surgically resected patients? And so I think this is good supportive data, and I ultimately think that these MRD assays will likely, number one, help inform adjuvant therapies, and two, might actually help us decide how long to continue adjuvant therapies for certain patients. So we'll see more data to come on that. The next study that came out last year that was really transformative was the LAURA study. The LAURA study is looking at osimertinib after chemoradiation. And so this took patients with stage 3, non-resectable but locally advanced EGFR-mutant lung cancer that underwent concurrent chemoradiation as definitive treatment without progression, either during or following chemoradiation, and then randomized patients 2-to-1 to receive osimertinib daily versus placebo. And importantly for this study, treatment was indefinite until progression. And so there was no set time frame where this adjuvant therapy was received, but really was indefinite until a patient progresses. Primary endpoint was PFS by BICR. And here you really see a marked benefit with the adjuvant osimertinib after chemoradiation, where the hazard ratio is 0.16, so perhaps even greater than what we see with surgically resected cases with adjuvant osimertinib. And you can see there's prevention of both distant and local progression with osimertinib versus placebo. One of the big issues or sort of points that were brought up with this study, though, is that this was a really high-risk group of patients. And so one of the concerns is only about half of patients got PET scans prior to study enrollment. And so a real question is this just telling us that osimertinib works for metastatic disease? Even when looking at patients that had CNS metastases, 10% had baseline CNS metastases. And so at least 10% had metastatic disease prior to study enrollment. And so I think that is a real question as to whether if there was kind of more standard of care, up-to-date staging prior to study enrollment, whether we would see such a market benefit. But one thing that I think it does raise, or a point that it raises, is, should we be giving osimertinib indefinitely for these locally advanced cases? And a lot of us were, brought up the question about whether for surgically resected disease as well, whether a prolonged or longer course of osimertinib would be beneficial. But I think that the as of now, take home from these studies is really that osimertinib has become the standard of care adjuvant therapy after definitive therapy for all locally advanced EGFR-mutant lung cancers, whether they receive radiation or surgery. The next compound that we'll discuss is patritumab deruxtecan. So patritumab deruxtecan is a HER3-directed antibody-drug conjugate. It's in the deruxtecan family of ADCs, which also includes trastuzumab deruxtecan and datopotamab deruxtecan, so all of them have the same chemotherapy payload. We've now seen data from several of these patritumab deruxtecan studies. So the first one is the phase 1 dose escalation study. And what was reported last year was the longer-term follow-up data from that phase 1 dose escalation. Since that phase 1 dose escalation, we've also seen the registrational phase 2 study looking at a very large number of patients who received the recommended phase 2 dose of 5.6 mg per kg every 3 weeks of patritumab deruxtecan. And so we have established the response rate of around 30% and the median PFS of about 5.5 months in a large data set of patients with EGFR-mutant lung cancer after osimertinib and after platinum-based chemotherapy. So with longer follow-up of the phase 1 study now, with several years of follow-up, the main points are that the progression-free survival and the OS are updated, but consistent with prior reports where we're seeing a median progression-free survival of 6.4 months, a median OS of 16.2 months. Importantly, as we've all been looking for biomarkers for these different ADCs, there was a retrospective look at HER3 expression and its potential correlation with response to HER3-DXd. And you can see here that there really was no way that the HER3 expression did not distinguish responders from non-responders, where there was completely overlapping HER3 expression among responders and non-responders. And I think part of the challenge is that all patients had some degree of HER3 expression. And so, I think there's more and more thought that actually very little of the protein cell surface antigen is required for antibody binding. And so I think one main point that you'll hear me say over and over is perhaps the patient selection or biomarker is actually oncogene driven lung cancer. And so a lot of these ADCs are now focused on EGFR mutant lung cancer that are kind of currently in development. Other findings from that longer follow-up is a suggestion that maybe patients with EGFR-independent resistance had higher response rates to pat deruxtecan. And so EGFR-independent resistance are patients that had MET amplification, KRAS alterations, acquired ALK fusions. I think just again reinforcing that these ADCs do seem to be effective amongst patients with diverse mechanisms of resistance to osimertinib. And then we saw the first data on acquired resistance to antibody-drug conjugates, specifically HER3-DXd. So one of the questions with antibody-drug conjugate drug resistance is, will patients develop resistance to the antibody component of the ADC or the payload? And I think the initial data from HER3-DXd really suggests both. So with sequencing of both plasma and tumor tissue when available of patients with acquired resistance to HER3-DXd, some of the alterations we found included a HER3 frameshift mutation that inactivated and kind of led to lack of dependence on HER3 signaling. And we actually also saw an acquired alteration in the TOP1 gene, which is the topoisomerase mutation, a gene. So I think that sort of explains to us that we can see resistance to both the antibody and the payload, and understanding a patient's individual resistance is going to be important, because if somebody has acquired HER3 alteration after HER3-DXd treatment, they still might respond to, say, trastuzumab deruxtecan because they don't necessarily have resistance to the payload, but actually the antibody. And so hopefully we'll see more and more data about acquired resistance to ADCs and how that should fit into clinical sequencing of the different ADCs. So in terms of where patritumab deruxtecan is in development, the confirmatory phase 3 study was a study that took patients after first-line osimertinib and then randomized patients 1-to-1 to either platinum-based second-line chemotherapy or HER3-DXd, with the primary endpoint being progression-free survival. We did see a press release back in September that stated that pat-deruxtecan demonstrated statistically significant improvement in PFS compared to platinum-based chemotherapy. So positive results via PFS. We haven't yet seen the full data, but this is encouraging to hear. And then in terms of where HER3-DXd is in development, in June of last year, we heard that pat-deruxtecan received a complete response letter from the FDA based on their BLA submission. And so what we heard was that the FDA did not have any issues with the efficacy or safety data with patritumab deruxtecan, but rather the complete response letter was a result of inspection findings at the third-party manufacturing facility. And so we don't have updated data as to when this will be resolved, but we do expect that patritumab deruxtecan will likely be approved in the future. So another agent, ADC, that we've heard a lot of data in the last year is datopotamab deruxtecan. So datopotamab is again in that deruxtecan family of ADCs, and it's a TROP2-directed ADC. And so one of the studies that we saw was results of TROPION-Lung05. And so this was a study that focused on actionable gene altered lung cancers. You can see from the pie chart on the top right, the breakdown of the different AGAs that were included in TROPION-Lung05, but really included patients with genomic alterations that received targeted therapy, but also that received cytotoxic chemotherapy as well. And so in total, 137 patients were treated. The response rate that was confirmed was 36%, with a median PFS of 5.4 months. We did get to see some later CNS data from this study, and really, illustrated that patients with and without brain metastases did respond to datopotamab deruxtecan, with a lower response rate in patients with brain metastases, and that possibly is representative of more aggressive biology for patients that had brain metastases. But importantly, there was intracranial response. The overall intracranial response rate with datopotamab was 22%. And really important to understand CNS efficacy in this patient group, because as we know, in particular with EGFR or say ALK positive lung cancer, the cumulative incidence of brain metastases exceeds 50%. And so we really need to understand CNS efficacy when considering or choosing between these different ADCs. We also saw combined data from both TROPION-Lung01 and 05. So this is combining really all patients with EGFR-mutant lung cancer that received datopotamab deruxtecan. And so when pooled together, that total was 117 patients. They really had the standard kind of clinical phenotype of patients with EGFR-mutant lung cancer, never smokers, Asian and white background, and somewhat younger age. The breakdown of the different types of EGFR alterations in that pooled data set is visible in the top right. And then the outcome data or efficacy data is right on the bottom of the slide, where you can see the confirmed response rate around 42%, 43%, median PFS of 5.8 months, median OS of 15.6 months. So far what we've seen, in my opinion, is pretty comparable kind of efficacy data with both datopotamab and patritumab deruxtecan within EGFR mutant lung cancer. I think the side effect profile is a little bit different, and I think is important to note. So all of these ADCs have the typical sort of cytotoxic chemotherapy toxicities, including fatigue, nausea, vomiting, alopecia. One difference with datopotamab is we're actually seeing a higher incidence of stomatitis or mucositis, which was present in 69% of patients. And then there are some also ocular surface events, so eye toxicities that are grouped together in about a third of patients. One thing to note, both the drug-related ILD independently adjudicated for both datopotamab deruxtecan and patritumab deruxtecan sits right at about 5%. So certainly present, but not as high as what we see with trastuzumab deruxtecan. And so where are we with datopotamab deruxtecan? So we just saw in November, so 2 months ago, that datopotamab deruxtecan submitted a new drug application, a BLA, for accelerated approval for patients with previously treated EGFR-mutant non-small cell lung cancer, so the similar indication as patritumab deruxtecan. And then in terms of ongoing studies, there are two important ongoing studies in EGFR-mutant lung cancer with datopotamab. The first one is a first-line study where they're looking at osimertinib monotherapy versus osimertinib plus datopotamab deruxtecan in the first-line setting. And so this is escalation of therapy that, if efficacious, would compete with osimertinib chemotherapy and amivantamab lazertinib as kind of escalated combination therapy. And then there's a confirmatory phase 3 study that is looking at datopotamab monotherapy or datopotamab with osimertinib versus platinum-based chemotherapy. And so looking at, does osimertinib add anything to the datopotamab, and then comparing to standard of care second-line therapy, which would be platinum-based chemotherapy. And both of these studies are ongoing but early in the stage. So what conclusions can we make from these different ADCs? I think one thing that seems to be an overarching theme is that perhaps the biomarker for some of these ADCs are the AGA population, where you can see that there is enrichment of efficacy of both datopotamab and patritumab deruxtecan in EGFR mutant lung cancer. And then I think another important conclusion is initially when ADCs were being developed, they were thought to be bulky compounds that were large, and sort of there was a hypothesis that there would be limited CNS penetration. But I think the data that we've seen from both patritumab and datopotamab really suggests that there is CNS efficacy that's actually pretty similar to the systemic efficacy, where we're seeing kind of CNS responses that are very similar to the systemic response rate. And then with both datopotamab and patritumab, there are efforts to combine the ADC with the targeted therapy, so in this case, osimertinib. And so the idea is there any additive benefit with continuing the osimertinib in these EGFR mutant lung cancers that are still dependent on EGFR signaling? And so we would like to see the data of those combinations compared to single agent ADCs. And, following that same theme, there's sacituzumab deruxtecan, which is another TROP2 ADC. And two studies that are currently ongoing are TroFuse-004 and TroFuse-009. So TroFuse-004 is again looking at patients with different AGAs, focusing on EGFR mutant lung cancer, but also allowing the different other AGAs, including ALK, ROS1, BRAF, NTRK, MET, and RET. And it is randomizing patients 1-to-1 to receiving sacituzumab deruxtecan versus standard of care chemotherapy, so either docetaxel or pemetrexed. And the primary endpoint is a shared primary endpoint of PFS and OS. And TroFuse-009 is more specifically looking at EGFR mutant lung cancer and allowing both the typical exon 19 deletion and L858R, as well as some of the atypical EGFR mutations, and then randomizing patients after they've received EGFR TKI therapy to either sacituzumab deruxtecan or the standard second-line therapy of platinum-based chemotherapy. And again, the primary endpoint of PFS and OS. And so you see a trend here of all of these studies being looked at as later line therapy, but also kind of assessing themselves against platinum-based chemotherapy, moving up to potentially the second line. And so in terms of where we are with sacituzumab, the FDA granted breakthrough designation to sacituzumab deruxtecan, again, for patients with metastatic EGFR-mutant lung cancer. The initial data that we've seen showed a response rate as high as 60%. It's a small number in 22 patients treated with EGFR-mutant lung cancer and a PFS of 11 months. And so we'll see if that data stands, but again, showing an enrichment of activity of these ADCs in EGFR-mutant lung cancer. And so, in conclusion there to add, I think you can see a trend here where all of these ADCs are looking at maybe a lower bar or a higher efficacy point in the AGA space as an entry point. A lot of these agents, in particular, datopotamab deruxtecan, first they were aiming for a very broad population. If you remember, the initial studies were looking at datopotamab in all non-small cell lung cancer, adenocarcinoma, and squamous cell. And then as we see kind of waning efficacy, there is this thought to look at what subpopulations might be enriched for efficacy. And I think that, there is a concentration of ADC development here. And then, of course, there are also studies combining these different ADCs with immunotherapy for the non-AGA non-small cell lung cancer population. But I think another kind of summary point when looking at these ADCs is that we're going to need a biomarker to select these, which ADC would be better. Because, in some point in the future, we might have patritumab, datopotamab, sacituzumab for EGFR-mutant lung cancer, but understanding which patient might benefit from which ADC first and whether there is the ability to sequence among the different ADCs is going to be a question that we need to answer in the future. And so switching gears here and thinking about EGFR exon 20 positive lung cancers, one of the drugs that we've seen some data for is zipalertinib. So zipalertinib is an oral EGFR exon 20 drug. When looked at in exon 20, the response rate looked to be about 40% with a good median progression-free survival. But since that drug began development, of course, amivantamab, the EGFR-MET antibody, has been approved in EGFR exon 20 positive lung cancers. And so a real question is whether you can sequence the different TKIs, the oral TKIs, with the antibodies. And so is there going to be cross-resistance or is there the ability to sequence? And so one of the later zipalertinib studies is helping answer that question. And so it's taking patients with EGFR exon 20 positive lung cancer that have already progressed on amivantamab and looking to see if zipalertinib is effective. The primary endpoint was overall response rate and duration of response. The initial interim report reported on 30 patients who had gotten amivantamab. And you can see I would focus on the amivantamab only arm. So 18 patients got amivantamab followed by zipalertinib, and the response rate was 50%. If you remember, the monotherapy zipalertinib response rate without prior AMI was 40%. So really showing pretty comparable efficacy after amivantamab versus not. The other thing to note is that there were 12 patients that had received another prior oral EGFR exon 20 drug. That might have been poziotinib, mobocertinib, or investigational agents like BLU-451. And you can see that the response rate was lower, 25% in those cases. And I think that intuitively makes sense, where if you've gotten another oral EGFR exon 20 TKI and responded and progressed, you're going to see less efficacy with repeating another EGFR TKI. But I think, very promising to know that there is the ability to sequence. And now we have to go both ways, right? Where is zipalertinib better before amivantamab, or is the better sequence amivantamab after zipalertinib? But I think really encouraging data in an area where we have really limited options. And so in terms of ongoing zipalertinib studies, there are two to note. There's Resilient-2, which is looking at zipalertinib in the first-line setting. but also in patients with active CNS metastases after other EGFR exon 20 drugs. And so that initial data of zipalertinib post-amivantamab did come from Resilient-2. And then also looking at other uncommon EGFR mutations like exon 18 alterations like G709X or G719 or S768I, which is another subset which we don't know what the best treatment is. And then Resilient-3 is the registrational confirmatory study to look at zipalertinib and chemotherapy versus chemotherapy alone as first-line treatment for EGFR exon 20 positive lung cancer. And so this is similar to the study that looked at amivantamab with chemotherapy that led to the amivantamab chemotherapy first-line approval. But I think seeing whether we can utilize an oral drug in that first-line space, there's a lot of interest in that.
Just a couple of follow-up questions. Just getting back to the last thing you were talking about, zipalertinib.
Yeah.
I never exactly understood why mobocertinib got pulled off the market. I guess because it didn't work first line. But any reason pharmacologically or clinically to think that zipalertinib is going to be different than the prior TKIs that have been looked at, like mobo?
Yeah, I think that it was honestly solely a drug development issue where mobocertinib kind of made the bet that they would be better as monotherapy compared to chemotherapy, which was kind of a large step to begin with, because if you looked at the single agent data, the response rate was only 25%. So, you're really cutting it close as to whether you'd be better than chemotherapy. I think Zipa is doing a different strategy where they're obviously combining with chemo, right? If you can't beat them, join them, right? That kind of strategy. But I do think that there's going to be real interest. We need an oral TKI for exon 20. There's just plenty of people that either can't tolerate amivantamab or don't want it. And so whether it's sunvocertinib, zipalertinib, we need to get some of these approved soon, hopefully.
So one final question, getting back to Dato in patients with actionable mutations. Did Dato cause objective responses in people with mutations other than EGFR? I know there was RET and all this other stuff in the trial. Were there mutations, were there responses seen there? And is there interest in looking at that more?
Yeah, I think there were responses in all of the different AGAs. The response rates were lower. Like I remember the ALK positive lung cancer responses to Dato being lower. And so it's a strategy, right? Like they're focusing on EGFR because it's the largest subgroup and did seem to have the highest efficacy. But they did report, responses in the different AGAs as well.
The other thing is, in the bigger study, Dato had benefit with adeno versus non-adeno or squamous. Do you think that that's because of the actionable mutations in adeno, or do you think that the benefit in it's the other way, that the benefit is in adeno and the actionable mutations are along for the ride?
Oh, I think it's definitely the former, Neil. I think that there's clearly, like, for whatever reason, an enrichment in efficacy of these. They're all going for EGFR-mutant lung cancer, which I think is funny now. So I think that there is an enrichment there. And then I think a kind of negative enrichment where squamous cell lung cancer seems to be less responsive to these different ADCs. But yeah, no, I think that they tried for the broad indication, didn't hit it, right? And so then now focusing on, okay, what subgroup can we look at where that benchmark is met that we might be able to get approval?
So do you think that if they get enough patients, they would see significant benefit in ALK and these other mutants?
I think so. I think that, I guess I would say I suppose so, but I can't be sure. So I think we've looked at EGFR mutant lung cancer from the patritumab studies and really showed that almost all of those cancers express HER3. I don't think we have that data with some of the like RET or NTRK or ROS1, where maybe the HER3 expression is lower and the response rate might be a little bit lower. But I would expect there to be activity in all of the AGAs, just maybe a little bit attenuated.
Just trying to sort of see if there's any themes here. You have the issue that in general patients with actionable mutations don't respond to IOs, although people, I don't know that we know exactly why, but at least, you know, that seems to differentiate that. And then I guess there's some actionable mutations like BRAF, I don't know about KRAS G12C, that maybe are a little different. They maybe do respond to IOs. Like, do you think that BRAF and KRAS would respond to DATO?
Yeah, I think that you're hitting kind of the within AGAs, there's a separation of what I would call the never smoking related AGAs
Right,
and the other, right? I
right.
think BRAF and KRAS, there are some BRAF V600E, it's bimodal, right? Where we think about half of them are kind of like in the never smoking category, but then half of the BRAFs do have a heavy smoking history and most of the KRAS do. So I think that it has to do with the biology, right? Is this driven strictly by the oncogene, or is there smoking related kind of mutagenesis that leads to probably better responses to IO and less good responses to these more targeted therapies?
And just to sort of complete that, does that mean that you think more about using an IO in a patient with BRAF or KRAS?
Yeah, absolutely. I just had this case in clinic, on Friday where there was a BRAF V600E with a heavy smoking history and a PDL1 of 90%. And I
Wow.
think, for that person, I think pembrolizumab would be appropriate, right? I
Wow.
think it, so I think there's probably, not consensus. Like some people might give them dabrafenib, trametinib, but a lot of people would, I think, start with IO because, IO has that potential tail of the curve, right? Where there are people who have durable, even complete responses. And so, I think, and then for KRAS for sure, I would not, I think I wouldn't start with one of the KRAS G12C inhibitors. I'd do chemo IO first.
No, that case is really great because I thought you were going to hedge your bet by saying chemo plus IO, but you just Pembra, right?
Yeah, I mean, well, that I would say if I had seen them, I would have been more conservative and hedged my bets. But they got one cycle of Pembro on the outside, and then saw me on Friday, and I was like, let's just continue. So she's not having talks. But yeah, you kind of read it. I think I would have probably hedged, but not TKI.
So interesting.